Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about the activation of the alternative pathway. In the previous video lecture, if you guys remember, we talked the activation of the classical pathway, which is actually the part of innate immune system. Okay, now this alternative pathway is also the part of innate immune systems. So before starting this alternative pathway, here I am advising you guys that first of all you need to understand about the classical pathway. Okay, so once you get idea about the classical pathway, then the alternative pathway will be very easy for you. Why? Because this alternative pathway is matching to this classical pathway, but there is a little bit difference. Okay? So let me erase this one portion and let me start the alternative pathway. So let me write alternative now again this alternative pathway is the pathway of the complement system, and again this alternative pathway is the it play an important role in the non-specific amino systems, and it is activated against bacteria and fungi. So let me write fungi. And now this process is start when the bacterial cell, let's say this is the bacterial cell. Now this process is start when the bacterial cell are get internalized through ingestion, inhalation or injection. Okay, once it get internalized, then let's say they activate the immune system, and in immune system let's say they activate the humoral immune system or the humoral immune response, let's say they activate, and in humoral immune response let's say they activate the complement system, and in complement system they usually activate the alternative pathway. Now what actually happen, how this alternative pathway is activated? It is usually activated against the bacteria and fungi. In this case, one protein of the complement system is activated, which we call that C3 protein. This is the C3 protein, which is actually the complement protein. It is activated in this alternative case. Once it is activated, now it is not activated, it is inactive form, but it is just circulating. It is just present in the bloodstream, and it is just present in the tissue area or any area of the body, but it is not in active form. Okay, now the activation is possible when it bind with the water molecules. Now once it bind with the water molecules, or let's say the water molecule are mixed with this C3 protein, then it is activated. Okay, so we get activated C3. Let me draw a diagram. Now after that, what actually happen, this C3 molecule or proteins bind with the factor B. So let's say this is the factor B. Again it is not activated. Okay, here the water are just mixed and they will just facilitate it but not activated. Now when the factor B's bind here, still it is not activated. How the activation is possible then? The activation is possible when another factor is bind here. Okay, and that factor is called a factor D. Let's say here the factor D are bind. Now once the factor D are bind, then they will activate the C3 protein. Now what actually happen? They will activate not only the C3 protein, they will just activate the factor B. Factor B once activate, then this factor B is converted into two fragments. Okay, one is the one we call that B and the other we call that BA. Okay, both are released out. Then what actually happen after that? This is, this factor D is also removed from this area because they perform their job. Now after that, what actually happen? This bimolecular, as we get the bimolecular complex, because this factor D is released out. Now this bimolecular complex will activate more C3 proteins, And after that, the C3 proteins are then cleaved because this bimolecular is act as a hydrolysis. They can hydrolyze or they can divide the C3 proteins. Now we get more C3 proteins. Let's say here we get another C3 proteins. C3 proteins. Now let's say this is the C3 proteins. These C3 proteins bind with this bimolecular complex, and again it is divided into two fragments. One is the smaller and other is the larger, if you guys remember. One is the smaller, means C3A, and another is the larger, means C3b. The smaller is released out, which act as an anaphylactic toxin, and the larger, which act as an opsonization, which usually responsible in the opsonization process. This C3b is bind to this cell membrane area. Okay, now let's say here the C3b are bind. In the previous video lecture, if you guys remember, we talked that here are the C2A, C1, C4, all the proteins are needed which make a C3 convertase. But in this case, the C3 convertase are not formed. C3 convertase are usually formed by, now how the C3 convertase is formed, now pay attention here. In this case, in the previous video lecture, if you guys remember, we just draw antibodies here, and we usually says that in classical pathway IgG antibodies are activated, but in this case IgA and IgE antibodies are activated, and usually IgA and IgE proteins are released by this plasma cells. And this plasma cells, if you guys remember, we talked that it is it is matured by this B cell. B cell produce plasma cell. B cell is converted into plasma cell. Plasma cell then mature and they produce IgE and IgA antibody. This IgA and IgE antibodies are bind to the surface of the bacteria, where the antigen are present. Let's say the antigen are present at that specific area. Okay, after that, here the antibodies are binded. So let's say this bacterial cell is called by two antibody, and one antibody is called at IgE, while the other is called at IgA. Okay, after that, once we get C3b, this C3b is covalently bound with this bacterial surface. Okay, now let's say this is the gram-negative bacteria. So due to this lipopolysaccharide, this C3b proteins are bind. Okay, now after that, what actually happen? This C3b protein again bound with the factor B. In this case, they again bound with the factor B. So let me draw. This is again factor B. Now after that, this is act as an enzyme, and they can again hydrolyze more C3 proteins. So again C3 protein is activated. So instead this C3 proteins are bound to that area. Now this time the C3 protein are not cleaved by this one complex. They are cleaved by this complex. Okay. For the first time, the C3b protein, the only C3 proteins are cleaved by this one complex, bimolecular complex. Now after that, the C3 proteins are now cleaved into two fragments or divided into two fragments by this one complex. Okay. Now after that, again this, let me draw again. This is C3. Again we get C3. This C3 is again cleaved into two fragments, C3A and C3b. Okay, C3A act as an anaphylactic toxin, and C3b again it is bind to that specific area. Now this is acting as a C3 convertase. In the previous video lecture, if you guys remember, we talked that C2A, C4b, we have C3b, all these are acting as a C3 convertase. But in this case, the C3 convertase are the C3b, factor B, C3, all these are actually the C3 convertase. Okay, now after we get the C3b proteins, this C3b, we get a lot of C3b proteins. The C3 are binded and they are cleaved. Again C3 are binded and the process are continue until the bacterial surface are covered, not the whole surface but some of the bacterial surface are covered with the C3 proteins. So let's say these circles are representing the C3b proteins. But when the some of the bacterial surface are covered with the C3b protein, then they activate. In this case, the C3a activate the mast cell and basophil. Now after that, the mast cell and basophil contain granules. These granules are fused, and in each granules there are the histamines and serotonin. Histamine and serotonin will dilate the blood vessels, and after that the other phagocytic cells are attracted. Now they can also cause the smooth muscle contraction. Now after that, once the phagocytic cells are attracted, so then this phagocyte cells can engulf it. This bacteria are then engulfed. But sometime what actually, sometime this one phenomena happen. Sometime what actually happen, this C3b will just bind to this. Again, they will bind to this complex. So let's say this is the C3b. Now again, this C3b binds to this one complex, and now this one complex is acting as a C5 convertase. So after that, C5 proteins are activated. Now once C5 proteins are activated, they are again cleaved by this one convertase. Now once it is cleaved, so again let's say the C5 protein. Let me draw C5 protein with the green marker. So let's say C5 proteins are activated. This C5 protein are again cleaved by the by this tri or they are usually tri or tetra molecular complex. Once it is cleaved, so then it is cleaved into two fragments. One is the smaller, other is the larger. The smaller is the C5a, which is again acting as an anaphylactic toxin, while the larger is the, let me draw here. So the larger is the C5b. Okay. Now this C5b is just circulating around this bacterial surface. They not attach. Okay, remember, they not attach. Now how the attachment is possible? What actually happen? Sometimes this C5b, a lot of C5b proteins are circulating. Due to this way, this bacterial cell are again looking tasty for the macrophages and dendritic cell or other phagocytic cell, and then it is engulfed by this C5b protein also. Now sometime this C5b will activate some other proteins which we called it C6, C7 and C8. First of all, C6, C7 is activated. So let's say this is the C5b protein. Now C5b proteins are not attached, but this C5b protein are just loosely bind. Not bind, but they just attaches the cell membrane loosely. Now how they are bind, how they are connected to the cell membrane? They will be connected when other proteins are get activated, and these proteins are the C6, C7. Okay. Now when C6, C7 are connected with this C5b, so then they will covalently attach this C5b. Not only C5b are attached, so these two proteins are also attached to this cell membrane of the bacteria surface. Now after that, what actually happen? They will activate the C8. This 3 complex will activate the C3 proteins. This will activate C8 proteins. After that, we will get the C8 proteins. Now if you guys remember, in the previous video lecture we talked that C8 is acting as a transmembrane activity. They will embed the whole complex C5b, C6, C7. They will embed each of the protein. Now once each protein are embedded, so a very small hole is created, not the large hole. This hole is facilitated and developed and become more larger by another protein we called it C9 proteins. So after that, let's say this is the C9 protein. After the C9 protein is activated, and they make the holes. these holes are usually formed at that specific area. The hole are usually formed at that specific area, but actually I draw here. Okay, now once the holes are formed, then what actually happen? If it is any eukaryotic cell, then what actually happen? This hole will absorb water molecules and sodium ion. So let's say this is the sodium and this is the water ions as you are watching. They are get internalized, the bacteria are swell up and finally burst or die. Now, if this is, let's suppose this is the bacteria, and let's say if this is the gram-negative bacteria, then what actually happen? In this case, what actually happen? Let's say this is the gram-negative bacteria. All of you better know that in case of gram-negative bacteria, they have outer membrane and they have inner membrane. Between the outer and inner membrane, they have the peptidoglycan, which is very thin peptidoglycan layer. Now, let's say the outer membrane is cleaved by each of the complement proteins, or not cleaved. This outer membrane, there is a small hole created in this outer membrane. Now, after that, not only one hole is created. Each proteins are not present only at that specific area. The same proteins are present at that area, at whole part or at whole bacterial surface. The same proteins are present. So we can say that a lot of pores are formed. So due to a lot of pores are formed, we can say like that. Now, if it is gram-negative bacteria, so then this gram-negative bacteria receive the lysosome. Now, after that, once they receive the lysosome, this lysosome will digest the peptidoglycan. So once the peptidoglycan are digested, so weak cell wall is formed. Due to this way, the cell are burst and die. Because all of you better know the cell wall is the protective layer. The cell wall is the very, it is the protective barrier for this bacterial cell. So when the cell wall are weak and when the cell wall are digested, so how this bacteria are served? The bacteria are finally die. So we can say that again, this one complex is acting as a make. Again, these one complex are what? These are the whole complex are the make. They can make membrane attack complex. Okay, now what actually the C5a do? In the previous video lecture, if you guys remember, we talked that C5a is acting as a chemotactic factor. So again, this C5a, let me draw here. This C5a, what they will do, they will acting as a chemotactic factor. Let me write chemotactic factor. What does it mean? It means that they will activate the macrophages and the neutrophils. Now, macrophages and neutrophils once activated, so then they will engulf each of the cells. This is the idea about this C5a. Okay. In case of gram-positive bacteria, as all of you better know that they don't have the outer membrane, they have the thick peptidoglycan layer. So it is very difficult for the complement system to digest or to kill this gram-positive bacteria because they don't have the lipopolysaccharide as well. Now, in this case, what actually happens? Sometimes this complement system is activated against our own body, but it is not usually happen. It is usually happen in pathological conditions. Okay. So when the gram-positive bacteria are get internalized, so it is very difficult for the complement system. Then this gram-positive bacteria is killed by some other amino responses. Okay. Now there is another questions arise in our mind that which one is the C3 convertase and which one is the C5 convertase. So let me write in a very short form. Whenever this one complex is formed, let me write, whenever this C3b and bb, now this complex is acting as a C3 convertase, and whenever this one complex is formed, let me write C3b, bb and C3b. Now this one complex act as a C5 convertase. So this is C3 and this is C5 convertase. So that's in a sense about the alternative pathway that how the alternative pathway are activated and how they can kill the bacteria and how the pores are created to this alternative pathway. Okay, I hope you get idea about the alternative pathway. If still you have any questions or if still you have any kinds of problems related to this topic, so you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, so please hit the like button, share this video, and stay tuned, because in the next video lecture we are going to talk about MBL pathway. So must subscribe the channel to get the updates of the more complement system, means MBL pathway. Okay, I hope you understand. Thank you so much for watching this one video.